So first, maybe I'll thank Jay and Sham for kicking things off and I think introducing some of the work that CZI is doing. And maybe just to provide a little bit of context, I don't have a ton of time, and so what I'm going to focus on really is specifically the Chan Zuckerberg Initiative's efforts within single-cell biology and the Human Cell Atlas. And so would be more than happy to talk about other efforts that we have going on. Okay, so would be happy to talk about some other areas that we have active at the break or throughout the next couple days. Okay, so maybe just the one first thing to frame is that CZI is a relatively new philanthropy. So now just around 3 years old. And the thing to highlight is historically there are two approaches to support science within the ecosystem, and people are probably familiar with many examples of each of these. So the first being grant-making foundations. So in the US, this would be organizations like the NIH and the Bill and Melinda Gates Foundation, internationally, the Wellcome Trust, et cetera. And then research organizations, so A*STAR and the Broad Institute and RECON and others. And CZI is a little bit of a fusion of both of these. And this includes active grant programs. You've already heard about some of those in the last couple talks. And then also some efforts internally where, based on and inspired by the work of our grantees, we're looking to build specifically open source tools to help their work more efficient and easier. And so in this context, we have in-house software engineers and computational biologists that are working with or collaborating with the community. And so this is actually the area where maybe I'll focus on in this talk a little bit more, and then at the end talk about some of the upcoming and current funding opportunities. So I'll kind of move right to left here. Okay, so as I mentioned, I'm not going to talk broadly, just in the interest of time, about all of CZI's efforts, but really focus on the support of the Human Cell Atlas, because that's why we're all here and what we're excited about. And so to kind of help put that in context of the organizational goals, here we're back to the Venn diagram, and I'm going to give a number of really, really brief snippets, and also then try to encourage people to reach out either to myself throughout the talk or throughout the rest of the conference, or to the individuals that I think you'll hear a little bit more about each of these throughout the next couple days. So the first is we are funding researchers worldwide, and hopefully you get a sense that when we say worldwide, we really mean very internationally, so we don't have constraints of a specific governmental or national funder, and so we're really excited by and eager to support international collaboration. I think IDA is a great example of this. We are helping to build a data coordination platform to house and to help the community access and use all of the data. So we refer to this as the DCP, and this is a collaboration that, again, I'll come back to, and there's many members of the DCP team here, and I think will be great for people to touch base with. And then finally, we are building various tools, and many of these are more in the category of analysis tools for single-cell biology, and appreciating that this remains a bottleneck. And then finally, the thing that I would be remiss not to mention is that CZI is by no means the only funder that's active in this space or actively supporting the Human Cell Atlas. And just to kind of highlight a few at the bottom here, including Wellcome and the Helmsley Trust, NIH, the European Commission, Wallenberg, German funders, I think that there are many, many funders, much like ourselves, that are really excited to support this work. Okay, so what I'll do in the next couple minutes here is just go through, and as I mentioned, just give very, very brief snippets and snapshots so as to hopefully frame and introduce some of these projects, and also encourage discussion and questions afterwards. And very, very well caffeinated, so I apologize for either being really quick or overly enthusiastic, but I am generally tend towards overly enthusiastic. Okay, so I'm going to tell you a little bit about the various grant programs that we have in the past. As I mentioned, I'll end the last couple slides with future grant programs. I'll introduce the DCP, and then two of those analysis tools that I mentioned called Starfish and cellxgene. So going back about 2.5 years, there were the first two RFAs from CZI Science, period. And these first two RFAs both supported single-cell biology in the Human Cell Atlas. And they were one-year pilot projects to help us start to get to know the community, and also to help the international community start to form around and create a foundation for this work. And in particular, the two kind of broad themes were, one, a series of pilot projects focused mostly on tissue processing and experimental methods and benchmarks. And then the second, on the right, was a pilot, or a series of pilot projects, 85 projects, focusing specifically on computational methods. And you can get a sense for the variety of the computational challenges that this community, I think, faces, from very, very core infrastructure, data compression, scalable workflows, to then some of the more specific questions that would influence and help the queries and the way that groups will use the atlas that I think Jay framed very nicely with the slides from Sarah Naviv, in things such as how to think about cell states and type and trajectories and other facets. So these two efforts then translated into what's really an integration of these one-year pilots into larger three-year pilots that we've referred to as the CZI seed networks to support the Human Cell Atlas. And these launched just earlier this year, kind of midsummer. IDA is one of 38 different projects. All of the projects are on our website. I would encourage people to go check them out, get a little bit of information on them, as well as understand who's involved. Great way to get in touch with those folks to the extent that you're interested in collaborations. And as I mentioned, and as Shem already kind of framed, this is a very, very large and international effort. And I think in many ways encapsulates and captures a lot of what we're excited about and a lot of the enthusiasm that the Human Cell Atlas has generated. So there's over 200 labs working in 20 different countries across 10 different organ systems on these projects over the next 3 years. And we hope that this opportunity, along with working with many of those other funders that I mentioned, are really going to pave the way towards the first draft and provide some foundational and benchmark data sets. Okay, so the other thing that folks that are familiar with CZI might know is that we are really, really proud and adherent to this kind of second to last little blippet of open science. And so in both the pilot projects and now these seed networks, there's a really strong emphasis on all of the derivatives and artifacts and things that are coming from the projects being openly accessible so that it's not just useful for the labs that are generating them, but for the community at large. So a few of these areas that I think have really taken off within the context of the Human Cell Atlas and single-cell biology include protocols. And this slide kind of grows constantly. I think when we saw Jay's earlier, we were maybe just under 100 protocols. This is probably even out of date. So it's a really, really active community. I would encourage people to use it both to share their protocols, but it's also a great dynamic way to fork and evolve and change protocols depending on what your interests are, as well as to start active discussions. And I think a few of the protocols here, including Steve Henikoff's cut and run, seem like there's just kind of a constant thread of discussions where groups are reaching out in real time and kind of trying to understand the ins and outs of new methods. So it's a great resource to understand various single-cell methods. The other thing that I would highlight is that it's useful, I think, more than just for experimental methods. So we saw the example of MuxSeq and demuxylate that Jimmy Yi's lab has provided. That protocol is here, but you can also use it for computational protocols, basically a tutorial. And so that's a really nice example because the computational method of MuxSeq is also on protocols.io. And so it's kind of a new, less represented and nascent area, but would encourage people to look at this as a great way to understand what's going on and how to move forward. Okay, so let me now very, very briefly touch on the data coordination platform. So the DCP was really the first grant that CZI made within the Human Cell Atlas. And it's a special and unique one because it's both very central to this community, but also it's a great example of that middle of the Venn diagram that I mentioned, which is where we are both funding the DCP, but we're also collaborating. So the funding includes support for the Broad Institute, EBI, the University of Santa Cruz, and Stanford University. But in addition to that support, software engineers and computational biologists at CZI are actively working with these organizations to create a robust and scalable and open platform to house the data that will comprise the Human Cell Atlas. And this community and this team has made a lot of progress. You can now, I think this got cut off a little bit at the top, but data.humancellatlas.org. The best way to get to know the DCP is to start interacting with the DCP, and I'm excited to say that there are many, many folks here from the DCP, and I think they'll be available at the break. And so maybe I'll skip over this and encourage everyone to go find the wranglers and the engineers and others from the DCP to learn more about and also to share what you're looking for from the DCP. Okay, so now let me shift gears a little bit and again, working left to right, get into the right side of the Venn diagram of a few small analysis tools that CZI is actively working on a little bit more in-house or also in collaboration. So the first one, Starfish, actually came from one of the grants in that very first RFA. And I mentioned that the primary goal of that group was to benchmark a variety of different methods. And one of the areas that I think many people in this room, as well as internationally, are really excited by, but also represents a huge challenge and really would benefit from some of this benchmarking, is the field of image-based transcriptomics. So this is a suite of assays where you use, in most cases, multiplexed fish and are looking at mRNA expression, but in situ in tissues. And there are a number of different methods. A few of them are here on the slide, but MERFISH, SMFISH, SeqFish, XFish, AwesomeFish, and so on. And in the spirit of trying to think about analytical roadblocks and how to help enable not just the groups that are developing these methods, but individuals and labs that would adopt these methods, a team of computational biologists and software engineers at CZI started thinking about a single kind of streamlined analysis pipeline that they termed Starfish. So Starfish, the goal is to help standardization and also adoption of image-based transcriptomics workflows. The approach is to think about a kind of unifying principle that really all of these methods are looking to do. You segment cells, you need to do some sort of spot detection to count your fish signal, and then ideally output a standardized count matrix, much like you would see with RNA transcriptomics. And so this is the team at the bottom here. They're working very closely with that benchmarking effort led by Ed Lean, and it's called SpaceTX. And I think there's been a number of different papers and preprints and things that are coming out and will continue to come out, as well as the raw data from all of these experiments. Okay, so once, for example, with Starfish or the ever-growing amount of transcriptomic data, another bottleneck or challenge is the data sets and these count matrices continue to get larger and become increasingly difficult to analyze and visualize and explore, right? They're extraordinarily rich with information. And so the second example of an analysis tool that CZI has been working on, and again, I should say that all of this is open. The GitHub repos are at the top. Would be happy to help make connections with anyone that's interested in using or learning more about these. So back to the visualization challenge. So the second tool that I'll talk about is a project called Cell by Gene. And Cell by Gene is not an analysis solution, so it's not looking to replace or change the analysis that many people are probably doing in Bioconductor or Seurat or ScanPy, but rather to take count matrices and just make it really intuitive and really easy and really fast, more than anything, to visualize these methods. And so Cell by Gene here, again, is a team primarily of front-end designers and visual designers, and then fantastic software engineers and computational biologists that are trying to think through how individuals are looking to extract information and analyze single-cell data sets and make that as easy as possible. It's a really, really fun tool. There's a couple great portals that have been set up for individual data sets, and again, would be happy to highlight this or help others find it. Okay. So I've covered a lot of ground with the single-cell biology, and now maybe I'll finish and we can all get to coffee with just talking very, very briefly about current and future funding opportunities. And in this case, I'm actually going to include single-cell biology, but then also more broadly. And so for some of these, again, would be happy to talk at the break. So the first within the single-cell program is related to, but distinct from, the Human Cell Atlas work that we've been funding. And this is focused on inflammation, so very broadly scoped. But the goal here is an appreciation that in many, many of the organs that people are, or that all of us are interested in as they go from homeostasis to disease, it's a challenge that involves local immune cells interacting with, in a dynamic way, the native tissue environments that they reside. And so the goal of the single cell analysis of inflammation, RFA, is to use these tools and technologies that just highlighted and that the Human Cell Atlas is leveraging to construct a reference atlas, to start thinking about how these interactions are mediating homeostasis or then perturbed either acutely or chronically in inflammatory processes to lead to common and rare disease. It's late, but it's not too late. So the application window here closes on November 19th. The one thing that I would say maybe as a word of encouragement is this is a very conceptual RFA, so we're really looking for ideas that connect various ideas within inflammation. And along those lines, it's a very short application. So hopefully, if you hadn't heard about this, there's not a lot of time, but enough time to hopefully pull something together. The next two, which may be of interest to many here, and there's more time. So the first is related to neurodegeneration. And this is an extension of our program called the Neurodegeneration Challenge Network, which I didn't talk about earlier. But it's very similar to, in some ways, the single-cell analysis, but it's looking to build pairs of researchers to explore high-risk and interesting domains of neurodegeneration. And then finally, a program that is now a rolling program to support essential open source software. I think this is probably of interest to many in this room, either as users of this software or as developers. We just announced the grantees of the first round, and then a second round for this program, which is really focused on maintaining and developing existing tools that many groups use, will be opening in mid-December, and then the third round will be open in 2020. Feel free to reach out or ask me more questions about any of these. Okay, and with that, I think I've managed to not go too far over time. Here, my email address is jcool@chanzuckerberg.com. Please reach out, my Twitter handle, as well as all the information if you're interested in any of our grants or tools or anything else. So with that, thank you again.